From root and stem, now let us move on to the next part of the plant, that is a leaf. Now we are going to consider both the dicotyledonous and monocotyledonous leaf individually, and this lesson aims at making you aware of what lies inside a dicotyledonous leaf. First thing that you have to remember, that we are talking about a leaf which could belong to a plant like a sunflower, it could be a mango plant, and such leaf would otherwise be known as dorsiventral leaf. Alright, now this term dorsiventral leaf is made with reference to the location of stomata. Alright, we are going to come to this term, but for once let us understand that what lies in a dorsiventral leaf and how are we seeing this leaf. We are seeing the vertical section of a dorsiventral leaf. Now when I say vertical section, you must be aware that if supposedly I am holding it like this is a leaf, this would be this would be the leaf supposedly, and this is the structure of leaf. This is how leaves are present. See, like this. This is a leaf. You take that leaf in such a way that it appears to be a sheet in front of you. You have to see it like this. Instead of watching it like this, if supposedly you're watching it from overhead, you're seeing the upper surface. If you're looking from below, then you're seeing the lower surface. What you have to observe is that what lies between the upper surface and the lower surface, and that section would be known as the vertical section of the leaf. Now supposedly this is the leaf, you're seeing the upper part, the lower part, and what lies between these two parts. So in that way, the vertical section has three parts which are to be studied. First one is epidermis, and unlike the previous epidermises that we did with in the case of root and the stem, where we had the epidermis which was circular, like it formed the circumference, the epidermis over here would be upper epidermis and lower epidermis, like this. Supposedly this is the upper epidermis, this is the lower epidermis, in between you find the vein, and the leaf would be subtended like this, something like this, like it. You are watching the leaf, this is the rib, you have cut it and you're observing it. This is what you have to observe. The leaf would be observed in this way. There would be upper epidermis, there would be lower epidermis. So in the case of epidermises, we have two epidermis, upper and lower. Alright. Now upper epidermis and lower epidermis, we are going to come to it, and the second one is the presence of mesophyll tissue. The part between the upper epidermis and the lower epidermis is known as mesophyll. So this particular part is mesophyll. As you can see over here, this upper one is upper epidermis. By now I hope that you get an idea how we are seeing the leaf. You have an idea from your understanding of the very first lesson where it was shown how do you make sections. This is the lower epidermis, and in between lies the mesophyll. Now two things that we have to discuss are considered. Moving further, what do we have next is the vascular bundles. So the vascular bundles would of course be xylem and phloem, and where they are located, let us see. Xylem and phloem. This is the vascular bundle which is surrounded by the bundle sheath cells. So we have vascular bundles over here. So in this way you represent a dorsiventral leaf, that is the leaf of a dicotyledonous plant in its vertical section. This is the way of representing diagrammatically a vertical section of dicot leaf. Now what you have to understand, that in the case of upper epidermis, the upper epidermis is having a single layer of cells which have chloroplast in them. The single layer of cells is covered towards the upper side with a thick cuticle. So we have a cuticle layer which is thick. Now because we are talking about epidermis, so we have to see that what is in store with the lower epidermis. The lower epidermis is also having a single layer of cells, closely packed cells, but we find firstly that the cuticle over here, which covers the lower epidermis, happens to be thin. Okay. Secondly, one point of consideration over here is, while the upper epidermis might be or might not be continuous, the lower epidermis is having pores in it. That there are small spaces which are known as stomatal pores. These small openings are the stomatal pores, and because we are taking into consideration a dicot leaf, the stomatal pores are in abundance on the lower epidermis. There would be many stomatal openings on the lower epidermis. Comparatively, upper epidermis has few or least or none of the stomatal pores present in it. So this is all about epidermis. Now moving further, we have mesophyll part that lies between the upper epidermis and lower epidermis, and what you have to understand in the case of mesophyll, that mesophyll is of two types. One is palisade and the second one is spongy. Now, when we say that palisade mesophyll, it is the palisade parenchyma, this particular part where you can see that cells are vertically arranged in close proximity to each other. It could be single layered or it could be double layered. So, we are having this part known as palisade parenchyma. Alright. This part is palisade parenchyma, and this particular part where you can see many cells other term is palisade mesophyll, spongy mesophyll, or you can say palisade parenchyma or spongy parenchyma. What you see over here, that these places, these particular places, they are air spaces, they are empty. There is no presence of cells in these spaces. So we have air spaces present in spongy parenchyma. That is why it is called spongy, because you know, in a case of a sponge, you have a little bit air spaces, and so is the arrangement that we have air spaces between the cells. The cells are not in close proximity to each other, but as you can notice over here, represented by small dots, this particular representation shows that these both palisade cells and spongy cells are rich in chlorophyll and they perform the function of photosynthesis. Now you might ask that why palisade parenchyma is so cluttered, or you can say why the cells are so tightly packed. The reason is that the upper surface of the leaf is subjected to sunlight, and these cells have arranged themselves in a crowded manner in order to carry out photosynthesis so that they can take maximum sunlight. The lower part of the leaf that lies towards the lower epidermis, that particular part has spongy parenchyma cells, again rich in chlorophyll, but these part of mesophyll, these spongy mesophyll cells, they serve the purpose of air exchange, and from where the air exchange takes place, that is known as stomatal pore. As you can see, the air spaces are in continuation with these pores, and from these pores the gaseous exchange takes place. Now what sort of gaseous exchange? You know, the main gases involved in the plant's life cycle are CO2, that is carbon dioxide, and oxygen, and of course one more, that is water vapors. Okay, so the gaseous exchange takes place from the stomatal pores. Talking about the stomatal pores which are present on the lower epidermis, the lower epidermis has specialized cells known as guard cells. Another point to be remembered. Okay, the guard cells which are present over here, it would be lining the stomatal pores. So you're going to find guard cells lining the stomatal pores, and these cells play an important role for the exchange of gases. So there is one specialized type of epidermal cell known as guard cell present around the stomatal opening. We have spongy parenchyma, we have palisade parenchyma which is part of mesophyll, and we have upper and lower epidermises. Now coming to the last part, that is the vascular bundles. This part is xylem. As you can see, the circular part is xylem. The lower one, represented by blue color, is phloem. So what you see, the thing to be remembered is that the phloem lies lower to the xylem. Okay, talking about vascular bundles, the thing that is to be remembered is that vascular bundles would be conjoint, collateral. You can remember these terms if you cannot recall by watching the lessons of previous class, and exarch. This is the condition of vascular bundles. Firstly, they are conjoined, they would be present in close proximity. Secondly, they are collateral. And thirdly, they are exarch. With reference to the protoxylem lies towards outside while metaxylem is towards the center. So, we have exarch, collateral and conjoined condition. Of course, they would be close because we do not see any secondary growth in the leaves. And moving further, there is one point that is to be remembered, that the vascular bundles are surrounded by bundle sheath cells. So, this circular arrangement that differentiates the other part of the mesophyll from the vascular bundle is known as bundle sheath. So, we are going to quickly revise what we have studied in the dicotyledonous leaf. First of all, it is a dorsiventral leaf, dorsal and ventral surface. The ventral surface has more of stomata as compared to the upper surface. We are talking about a leaf of, say, sunflower. In the leaf, we would find three parts: epidermis, upper and lower. In between, we find the mesophyll, which is again divided into two easy distinctions. One is palisade parenchyma and the spongy parenchyma. As the name suggests, palisade is an English word which says that the arrangement is quite close to each other. As seen in the case of books, you would go inside a library and you would find that books are arranged after one another. That is palisade arrangement. So is in the case of the cells, we have palisade parenchyma, and lower to that we have spongy parenchyma, which is towards the lower epidermis, which is rich in air spaces and cells which have chloroplast. These air spaces are in continuity with the stomatal pores for air exchange, okay, exchange of gases, and these stomatal pores are part of the lower epidermis. The cuticle on both the epidermis varies in the thickness. While the upper cuticle is thick, the lower cuticle is thinner. Now, coming to the vascular bundles are present in the midrib, basically part of the leaf, and they are surrounded by bundle sheath cells. In the vascular bundles, you are going to find xylem above while the phloem would lie below the xylem. The arrangement of the vascular bundles is such that we can term it as conjoint, collateral and exarch. And this is all that you have to learn in the case of dicot leaf, and do practice making the diagram so that you get a better hold of all the points that we have studied.